Hello everyone. Welcome to today's session, today we have the 5 selfish reasons to work reproducibly, one of our seminars, Future Forward, Science in the Open Era. And I would like to welcome you all. My name is Marta Teperek and I'm from TU Delft Library. I'm working as the Data Stewardship Coordinator. And I would like to welcome you all on behalf of the entire library and all the people who helped us prepare this event today, including our colleagues from the communication teams that worked really hard with us to make sure we have nice feedback forms and nice advertisements of some other events which are upcoming. And also people who helped us with the catering that you will have afterwards. So welcome to our event organized by the library. And just to tell you, why do we bother? Why do we organize Future Forward seminar series? Why do we care about open science? So in general, probably you've heard a lot about open science, about the new requirements, about funders, asking researchers to share their research openly and so on and so forth. But we thought it's useful to discuss those topics. There are some difficulties, there are some challenges. What does it mean to work reproducibly? What does it really entail for every researcher in their daily workflows when working on the research projects? So that's why we started those discussions as a place where you can really start to have conversations, where you can share experience. And importantly, everybody is welcome. So you don't have to be already a keen person who is practicing open science. You don't have to know all the answers, how to work reproducibly. That's really a place where we can ask questions and exchange experience. So hopefully there will be lots of opportunity for that and discussion as well. And finally, before we start, I would like to know a little bit more about you, if possible. So I have some questions for you to get us started, get us going, and get us discussing. And the first one is, we would like to know, who are you? So if you can please raise your hand if you are an active researcher. Perfect, thank you. And can you please raise your hand if you are research support staff? Thank you. And who didn't raise the hand yet, or who is other? Okay, thank you. And another question for you. Who is from TU Delft? Please raise your hand. Thank you. And who is from outside of TU Delft? Thank you very much. And the final question for you. Can you please raise your hand if you have attended any of the seminars in this series before? Thank you. And can you raise your hand if it's the first seminar in that series that you are attending? Thank you very much. That's all the questions I had from you. And just some practical notes before I start. So you probably noticed we have a video recording today thanks to colleagues from Collegerama and also from the New Media Center. And what we will be doing, we'll be recording today's presentation, so the speaker will be recorded, but also the questions from the audience. We would like to make this talk accessible to people who couldn't be here with us today. So a couple of things. If you don't want to appear on the recording, please don't ask a question during the Q&A. You can do it later on in person. And please make yourself known to the people doing the video recording in the back and in here to make sure that they don't take the recording of yourself. And one more thing also, because we do the video recording, when we do the question and answer session at the end, please always introduce yourself and make sure you speak to the microphone so that people can hear what you have said. And the very final thing, my colleague Sandra has prepared some wonderful feedback forms that you have probably all received. So before you go, just remember to give us some feedback on the event today so we can continue improving what we deliver for you. And without further ado, I just wanted to introduce you to the program of today. We are just finishing with the welcome remarks. We will have the talk by Florian Markowitz about the 5 selfish reasons for reproducible working. Then Rob Hoft from DTL agreed to chair the Q&A session and deliver some closing remarks. And afterwards, we'll have drinks reception. So please stay and use this as an opportunity to network, discuss, and also talk with the speaker. And of course, importantly, who is Florian? So I would like to introduce Florian Markowitz, who is the group leader at the Cancer Research UK Cambridge Institute. That's part of the University of Cambridge broader complex. He has lots of degrees, as you might see. He has a degree in mathematics and philosophy. Those two very often go together, as it seems, and also a PhD degree in computational biology. And his current research is focusing on cancer research, so very important to all of us. And he's working together with computational researchers and also with experimental researchers. So quite a nice combination of the two of those. And my personal note, I know Florian from while I was back at the University of Cambridge, a researcher. I've never seen his talk in person. I've only been fascinated by his talks on YouTube. So I'm actually quite much looking forward to Florian's talk today. And just as a spoiler, Florian already told us that actually his talk is not very reproducible. So apparently there are now more than 5 reasons to work reproducibly. And without further ado, I would like to welcome Florian and give his talk.
Thank you. Thank you very much. It's a pleasure to be here. I will entertain you with at least 5 reasons why you should work reproducibly. So I work in Cambridge. I work at the Cancer Research UK Cambridge Institute, and I work with people from all kinds of different fields, asking way too many questions. The only thing we have never been accused of is having focus. So part of the people I work with work on cancer genomics, so trying to understand the mutations in cancer genomes. Some of them work on the tumor tissue, trying to analyze histopathological, radiological images. And some of them do experiments perturbing genes and looking at how cells change and trying to understand how the internal wiring of a cell works. The people I work with come from computer science, statistics, biochemistry, medicine. What I'm telling you here today is what I tell all of them. So it's regardless of whether you're a dry lab or a wet lab person, what your background is. These are very basic concepts that I'll be introducing, which are applicable to every type of science. The key idea is there shouldn't be any miracles in science. You must have seen this cartoon before. You see these 2 elder gentlemen looking at a chalkboard. They apparently are mathematicians. There's a very complex equation on the top left, very complex equation on the bottom right. In the middle, a miracle happened. And then one of them says, I think you should be a bit more explicit in step 2, which apparently is the miracle. This is exactly how it feels to me when I read one of these really dense Nature papers, where people have done many, many years of research. There's a huge pile of data, which is somehow condensed into these 7 pages and 4 figures that they have. And unless you can trace back how the data were analyzed and how they made sense of the data between the original data and the paper, just a miracle seems to have happened. And that shouldn't be the case. Science should be transparent. There should be a track record of what you actually did. And sometimes it's not. So let me start with a case study, a story from cancer research. It's several years old now. It's called the Duke scandal. These 2 people here are Keith Baggerly and Kevin Coombs. They are biostatisticians at MD Anderson. Is anybody here a biostatistician? No. So it's safe to say this is not the sexiest type of science, biostats. These are not the heroes of science. These are not the rocket scientists, okay? And still, they made it to the cover of the New York Times. They made it there because they discovered that people in Duke had completely messed up. So the story is this, and these are slides I stole from Keith Baggerly. Many years ago, 2006, the group of Joe Nevins in Duke reported, and these are the really, if you're from biological sciences, these are the really cool journals. Nature Medicine, the New England Journal, that's the kind of the polished stuff you want to get into. They reported that they could predict from cell lines, these are cells that live on plastic, and by treating these cell lines with different drugs, they could predict whether a patient, a human person, would benefit from the drug or not. Well, there's a huge gap in between like cell lines, things on plastic, and the human being, so this was a major step forward. And these are like these genomic signatures to guide chemotherapeutics. Everything is cool. They made it to the big journals. They had one of these cool publications after the other. Obviously, people in other cancer centers got really interested, also at MD Anderson, and they said, listen, you biostatisticians, get the data, try to find out if we can get these signatures to work in our own research. And so that's what they did. Took them a while to get the data, they started to analyze the data, and they found lots of weird things. For example, so these crazy numbers here, they are identifiers for genes. What the people in Duke had said is, when we treat these cells on plastic, we find certain set of genes which react, and by looking at what genes that are, we look into a patient, we can predict what the patient does. So what the MD Anderson people did is they looked at there, so this is the list of genes that Duke had predicted, and they reanalyzed the data and they came up with another list, ours, the MD Anderson list, of what they had found. There was no overlap whatsoever. But there was a pattern. So look at what Duke had found. There was a gene called 1881 and MD Anderson found 1882. Duke found 31321. MD Anderson found 31322. So it seemed like there was no overlap, but they just found the same rows in the data matrix, just shifted by one. Why had that happened? Duke had used the devil's tool. They had used Excel. So what they had pasted, they had copy-pasted their data from an Excel spreadsheet into some statistical tool, and because they had to copy the data and they had to copy the row annotations, and the row annotation had a header saying gene names, everything was shifted by one. Terrible. This is not yet fraud. This doesn't end your career. This happens to everybody. This is in all my papers somewhere hidden. This is not a big deal. But it was just the beginning when the MD Anderson people were like, maybe we have to dig a bit deeper on what's happening here. Now, when you have a predictor, so you say, from your data, I predict something, you always need to prove that you can do this also on other data sets. So you need, on top of the data you build your predictor on, you need a test data set. So the Duke people also had test data, and they had found other publications where people had similar data than they had, and this other publication had, let's say, 50 patients, and Duke then published a paper saying, we took those 70. So the original publication from test data had 50, Duke, when they republished the data, claimed it was 70. So the MD Anderson people said, where do these additional 20 patients come from? So they plotted the data that Duke had published, and they plotted the correlation matrix of these different patients. And you will not be surprised to see on the main diagonal a correlation of 1, because everything is very highly correlated with itself. But there were also very high correlations, very close to 1, pretty much 1, on off-diagonal elements, which means there were pairs of patient data which look exactly the same. And if you have very good eyes, you see it in this data matrix. For example, here, all these columns look the same. All these columns look the same. There's even here, let me see if I find more here. All these columns look the same. So somebody must have copy-pasted all these patients in and just artificially inflated the test data. Why would you do that? Well, the more data you have, especially when you know how they look like, the better your accuracy will be. The better your accuracy estimates will look like, right? The people were not really smart, so sometimes they gave this, patients the same labels, good. Sometimes they gave them opposing labels, sensitive or resistant to the drug. So they were very bit sloppy on top of not being very honest. So these are just two examples of the flaws that the MD Anderson people found in the Duke paper. Now, because the Duke research had been so high impact, and because the story was so good and the journal names were so grand, Science and Duke had moved really fast, and within 2 years they had several clinical trials starting, some at Duke, some at Moffitt. This is really fast. 2 or 3 years for a clinical trial based on basic research is really, really fast. In one of these trials, the MD Anderson people found out somebody had switched the labels, so they were actually giving the drug to people that the classifier predicted for not being sensitive to the drug. So there were lots of mistakes in here. MD Anderson now started to communicate with Duke and Moffitt and said, listen guys, we read the papers, we reanalyzed them, there were lots of problems in here, we should talk. This is called the Duke problem or the Duke scandal, not the Moffitt scandal, because what Moffitt said is, they said, there's a problem, here's everything we have done, here's all our data, please talk to us, we fix this. Duke said, what are you, biostatisticians? Do you even publish in Nature? I mean, do we even talk to you? We just had 3 more Nature papers about this. Who are you even? This is the reason why it's the Duke scandal. The last author had to retire early, and the first author is now a convicted fraudster. So it was a big, big splash in cancer research. When Keith Baggerly came to Cambridge about 9-10 years ago, and he gave this talk, it was an hour of fun, we were all laughing so hard. Midway through the talk, half the audience stopped laughing. It was the smarter half. We were all sitting there thinking, shit, that can happen to us. Because how do you avoid little mistakes like a off by one? You don't. How do you avoid that the first author is a fraudster? You can't. But maybe you can check. And we all realized we didn't have all these checks in place, and we actually maybe we didn't really care. But something like this is a wake-up call. And there's many fields where problems like this have become apparent, in psychology, in economy. This is the story from cancer, the big reproducibility disaster that woke everybody up. Now, the first take-home message is, it's not all about fraud, my story. I mean, yes, the first author was a fraudster, but for me, this is a story about transparency. Many of these mistakes could have just been found by anybody who would have spot-checked the data. And many of these mistakes were so simple that you do not have to be a statistics wizard to find the problem. Seeing that one study reports 50 patients and the other study reports 70 patients is not wizardry. I mean, you just see the difference. Everybody can count. So the reason this blew up is that nobody saw the data. Just the first author, the person who manipulated the data, was the only person who ever looked at it. And these are data sets which are small. Everybody, nowadays you can analyze them on your cell phone, I guess. I mean, even then people would analyze them on their laptops. These are not big data sets. There's no difficulty in sharing them. It's just nobody did. And it's not all about the first and last authors. So the first author is a convicted fraudster. The last author was very famous and had to retire early. But there's many authors in the middle. Some of them have applied to the place I work at, and we looked at the CV, and we're like, this stinks. And there was that. So you can be caught in this too if you publish, even if you're completely honest, if you collaborate with people, if you never check the data yourself, you can be caught in a story like this. This is terrible. So when people hear these crazy stories about fraud and intransparency in science, they say, well, let's work more reproducible. Let's work more transparent. And why should we do this? Well, it's the right thing to do. It's the honest thing to do. It's the foundation of science. Reproducibility is the foundation of science. And we make the world a better place when we work reproducible. It's not that I think anything is wrong with those reasons. It's just, they're a bit preachy. I'm not a missionary. I mean, live and let live. You do your stuff, I do my stuff. I don't really care about making the world a better place. I mean, this is what these people do, and I don't wear sandals. I'm not in this game to make the world a better place. I'm in this game to win it. And when I look at you guys here, everyone here sitting here, you're winners. You're winners like me. You are in this game to win. So the question is not what you all can do for reproducibility, but what reproducibility can do for you. So here come 5 reasons for the selfish researcher who wants to have a good career and a longer CV why reproducibility helps you. The first reason is obvious after this introduction. You avoid disasters. I mean, you catch your postdoc who manipulates the data, or if you're a co-author, you see what's happening in somebody else's paper. You're just more aware of these problems. And the Duke scandal is big, but there are many smaller problems which can just be solved by storing your data in a way that you can find them again, annotating them, sharing them. For example, this is from Cambridge. Cambridge is apparently a very famous university, and people are in awe of how smart Cambridge students are. So this one is particularly smart. He had 5 years of research on his laptop in his backpack, and he lost the backpack when getting drunk in the Penton Arms. And then there was the only copy of his PhD. And then you found all through Cambridge these little flyers saying, please give me my backpack back. Terrible. This is a paper I found like a couple of years ago from Science. It's an editorial expression of concern. 2 researchers, Anna Lundstedt and Peter Eckloff, they are interested in fish eating plastic. I mean, that's what the Science publishes. The interesting thing in the expression of concern is this sentence. The authors have notified Science of the theft of the computer on which the raw data for the paper were stored. These data were not backed up on any other device nor deposited in an appropriate repository. So it's not just Cambridge students. I mean, it is people who publish in Science. I mean, I hate to say it, but Drs. Lundstedt and Eckloff, with this sentence, are Science certified idiots. I mean, if this is really what they did, they just stored it on one laptop, which then gets lost. Well, turns out this is also fraud. Apparently the first author never did any of these experiments. She just claimed she had done them and nobody could ever find it. But even, you see, the problem is if you act like this, if you don't have your data stored in a way you can find it again, if you lose your data, you look like them. You don't want to look like them. Now, it's a lot of fun to make fun of other people. Here's a story from my own lab. Several years ago, we were approached by collaboration partners. They did things called perturbation screens. So you're interested in particular cellular function. You knock out all the cells in the genome and you see when this particular function, when it vanishes or when it's active. So you find the genes which are essential for a particular function. They are called the hits. The problem is you never know how they work together in the cell. So I said, haha, I know what to do. We take some of these pathway members, so they work together in pathways, these genes. We take some of the known members, we take some of your new hits, we perturb them again, we do some particular readout, and I have a statistical method which puts it all together and gives you a graph of how everything is connected. And we did the magic, and this is called the NF-kappa B pathway, and if there's biologists in the room, I hope not, this is how the NF-kappa B pathway looks like. It's exactly what the textbook would say, and then there's the new hits coming in here. Beautiful. Our collaboration partners went to the wet lab, they started validation experiments, and pretty much this is the correct solution. So we thought we write this all up and send it to this journal that also writes about fish eating plastic. And we're very keen to submit to a high impact journal. So I say to the postdoc, listen, for submission, wrap up all the code, wrap up all the data, and so we are ready to submit. Nothing seems to happen for 2 weeks. After 2 weeks, he comes back and says, Florian, this is funny, you will laugh. I'm like, I don't think so. He's like, I just can't reproduce this graph. I'm like, what do you mean? I'm like, well, maybe the parameters of the method have changed, or the method has changed, or the data has changed, or I don't really know, but I get many other graphs that also look good. I was like, but this one is the one we validated. I mean, it looked like if you can't get to your hypothesis anymore, this is really when magic starts to happen. I mean, you say we somehow pulled an hypothesis out of thin air and then validated it. If we somehow lose the track record and the story, how we got to this initial hypothesis, the whole story is dead. And that was never published, was never submitted. This paper is just dead. It's very sad. This was years of work, our work and our collaborators' work, which in the end was lost because we had no clue how we ever got to this hypothesis. It was our fault. So what I learned from that is that a publication or any project is just more than a beautiful result. You always have to keep track of how you actually got there, because else magic seems to happen. It's also, if you start too late, you're screwed. So start early on with reproducibility. Maybe not on day one, you need to do some exploratory stuff, but as soon as you think you know what the project will look like, as soon as you know you have like your way in, the first results come in, start acting reproducible, keep versioning systems active, store your code, just, I don't know, keep a track record of what you're doing. It's not super hard. Because if you do, and if you actually make it to writing papers, reproducibility helps you again. It makes writing papers easier. And papers are the currency of science. They make CVs longer, and long CVs is why we're here, why we do science. So this is another example from my lab. It's a paper on cancer evolution, and it's in PLOS Medicine. When you look here on all this kinds of information you have about the papers, there is supporting information, and one of the supporting files is called a SWIF file. Nowadays we would call this an R Markdown file. This is a file that has text descriptions and code in it. So it looks a bit like this. There is R code, and there's also a little bit of text explaining what you see. If you push a button called Compile PDF, you get a nice PDF. And for example, this is the data table that we then later use to do survival analysis on. This is reproducible. So if you can just go online, you download this file. If you know how to use R, you can just run exactly our analyses, and you will get exactly the same figures that we have in our paper. Please don't try, because this is years ago, and I guess it's no longer working. But in theory, this is how it should work. And for us, this was really useful to do it this way. because initially some of our students and postdocs had analyzed the data, but in the end when we wrote it, the other PI and I sat there and we had to look at the data, we needed all kinds of information, and in this form it was all just really nice, easy to see, and every time we changed something in the data, all the analysis was automatically update because they were part of the code in here. So if I read in a different data file in the beginning, all the future analyses just changed automatically. So it was very easy to read, to get all the numbers we needed for the paper, all the p-values and hazard ratios and all of this. We just read it off the PDF. All the results automatically updated when the data changed. New patient came in, p-value got smaller, yay, we immediately saw it. Was very engaging because it was easy for us to read this and read the complete analysis. We didn't have to dig around in files and somewhere. Everything was well documented and explained. And it was easy for us to spot mistakes. So from the same collaboration, we were doing some survival analysis in ovarian cancer, and we were surprised that some of these survival analyses didn't come out the way we had thought. And we went through all the different parameters that we could think of, and we looked at tumor grade, which tells you how well developed or far developed the tumor is. And the stage actually. And what we were expecting were numbers between 1 and 4, with lots of 1s and fewer of the high stage 1s. What we saw in the data was numbers 2, 3, 4, 999, John, and XXX. So the person who had given us the data had done a really bad job at curating it. And for us it was very easy to find it because this was not hidden. This was just part of the data, was easily accessible, everybody contributing to the project could see it, and was very easy to spot where the problem was. The alternative would have been for us just to point at the postdoc and say, the survival plot looks funny, just do something. But this way we could actually do it ourselves. Now, when the paper got to the reviewers, reproducibility helped us again, because reproducibility helps you arguing with your reviewers. So this is the tumor evolution paper again. We had this markdown file, we had all our data accessible to everybody, including the reviewers, and one of the reviewers comes back to us and says, well, thanks for this, I downloaded the data, I downloaded the code, I played around with it, if I change the statistical analysis, it's no longer significant, what's happening? And we said, oh, that's very interesting, but the reason is this, and just use this other method like we did in the first place, and everything is cool again. Think about what happened there. This reviewer was completely on board already. This reviewer was not saying, oh, what a stupid story, should never been out there. Why care about this anyhow? We were having a constructive discussion about what the best statistical method was to get to the result. So it was technical, it was constructive, it was very detailed. And the only reason we could do this is because we had made all our data and the way we had analyzed it accessible to the external reviewer in a format that this person, who is actually a medic, found very easy to interpret and to manipulate. So we had a very constructive discussion with the reviewers, and in the end we got accepted very easily. Finally, back to your own lab. Reproducibility helps you with continuity. Think of this. You start new in a lab and you say, well, my boss wants me to continue the project of a previous postdoc, but that person left nothing. No scripts, no data. I mean, what do I do? Or maybe if you think about your own research, somebody comes and asks you a question. You say, well, I did this 6 months ago. How much do you remember of the details from 6 months ago? Well, in my case, would be very little. And many of us are so busy, they have many different projects. I mean, how much can you really remember all the details of all projects? Unless you have documented them well and you have stored them well in some format that is easily readable, and then you just look it up. And this is, again, for selfish reasons, because the first person who will reuse your data and your analysis will be you yourself in a few months, and your past self doesn't answer any emails. And finally, reproducibility is good. It helps you maintain and build your reputation. You get invited to famous universities like here in Delft when you talk about it. It's really good for your CV. So one way we do our reproducible research is through experiment packages in R on the bioconductor project. And so for some of our papers, it depends a bit on the size of the data sets. We have the data plus all the analysis scripts in one package. Like here from 2012, the Mulder package. You just download this package and you have all the data and all the scripts together and can directly reproduce all the figures from our paper. And we don't have this for one paper, we have this for more than one paper. So now in my CV, I have a whole list of software that my lab has produced and also data and other resources. So when people ask me for a grant application, what's your data management plan? Do you have a track record of sharing data? I'm like, long list. And long lists is what science is all about. Finally, there is a trend in many journals to actually become more transparent. So this is Brian Nosek's paper from, I forgot when, 2015 apparently, on the top guidelines for transparency, openness, and reproducibility maybe, I don't know. So what you see here is different types of features of papers like citations, data transparency, code transparency, and different levels of how top you are. Not caring at all to being completely open. Now, if this comes out in Science, this means all the top journals, the ones we all would like to publish in, are thinking about this. And if you follow simple rules of using R Markdown, for example, or these little experiment packages, you very easily find yourself in level 3 of these guidelines. And this is what very soon will be what is required. So if you want to publish highly, be reproducible, be in high level of the top guidelines. So those were the 5 reasons. You want to avoid disaster. You want to write papers. You want to talk to reviewers. You want your work to be continuous. And you need a reputation. That was that. But there's more. I've given this talk quite a few times. And people listen to my reasons. And then some of them say yay, because I often preach to the choir, and some of them just shrug their shoulders. So here are some of the responses that I got. One of them, that's actually from Cambridge, says, just because you do open science doesn't mean you're right. That is true. That is completely correct. It just means I do science in a particular way. It doesn't mean I'm more right than somebody else. It's just, I think, listen, you guys have dogs. If the dog comes in the house and shits in the house, do you want it on the rug or smeared underneath the rug? It's much easier to clean when it's on the rug. And that's the same here. If some problem happens, you want it to be seen and to be easily fixable. You don't want it hidden and smeared away like the Duke people did. Blah. Other people said, well, reproducibility is a bit nerdy, right? It's all for these nerds with OCD, not for real people like me. That's not how I see it. I think reproducibility is more about empathy. Because you have to think, to put yourself in somebody else's shoes and think, would that other person understand what I'm doing? Am I documenting, am I writing my notes in a way that somebody else can understand? This is not OCD, this is really empathy. Some people are very hard-nosed. They say it's only the result that matters. And I completely disagree. I mean, my story with this NF-kappa-B pathway shows that's not true. You might have a really nice result. If you can't tell how you actually got there, magic happened and you can't publish this. I'd rather do real science than tidy up my data. I have to say, if you don't keep your data and your code and everything clean, I mean, I don't think you're doing science at all. This one is a tricky one. We can always sort out the code and data after submission. You could, but nobody does. So before submission, early on is the right time to do this. People afterwards, imagine we had done this, we had submitted our paper on this pathway and then sorted out the code afterwards. We would have been in a really weird situation when it came to the revisions, because by then we would have known that we had no clue ourselves how we actually got there. What do you do then? You write this awkward letter to the editor saying we retract our paper before it's accepted? I mean, it's a bit weird, right? So you better sort this out early. My field is very competitive and I can't risk wasting time. I mean, true for all of us. I mean, who is in a non-competitive field? And this is why I think you should start early on with being working reproducibly, because you will save time later on. If in my NF-kappa-B example we had sorted this out earlier, we wouldn't have wasted 2 years on experiments. We would have just realized there was a problem early on. This way we wasted money and time. This is mostly for me because I'm a mathematician and I always have to fight with these wet lab biologists, and some of them say, well, if there's a wet lab validation, who cares if the computer stuff is reproducible? And I don't completely agree with this, partly because it assumes there's like a hierarchy in science where one stuff beats the other. So if you have like a real experiment with your pipette, it's somehow more important than my experiments on my computer. I do not agree with this. In particular, I think all parts of science should be reproducible. So I'd rather turn this around and say, how reproducible are your wet lab experiments? Are they documented well? What about your lab notebooks? Could I look at those and see what you actually did? Can you share them? Some people say this: Excel works just fine, I don't need any fancy R or Python or whatever. You've heard my opinions about Excel already. For some things it's good, right? When you gather your data and you need lots of manual curation, maybe that's what you want to do. But at some point you have to freeze your data and switch over to something that is scriptable, that can be easily read into a computer, where instead of clicking and pasting, remember the Duke people, one off, huh? That was Excel. Instead of clicking and pasting, you just write a script which just keeps track of all the things you're doing. And at some point, very early on, it's time to get rid of Excel and use a professional tool for data analysis. Mind your own business. I document my data the way I want. Maybe you don't like R Markdown. That's cool. Please do whatever you want. I mean, please just document it. The current reward structure does not enough to incentivize data sharing. I mean, I gave you 5 reasons, okay? What do you want? A pat on the back? I mean, there's enough out there. I mean, people claim, everybody blames the system. I don't want to do it because the system is against it. I mean, I just gave this 2 weeks ago to a couple of PIs in Cambridge, and I had to tell them, you guys are the system. I mean, who else is the system? I mean, if you review a grant and they don't say they share the data, don't give them the money. If you review a paper and the data's not in there, don't give them the paper. I mean, you are the system and you do your own incentives, so just do it. So, everything convinced? Still, there's lots of resistance to all this reproducibility stuff. So let me tell you another story. I do cancer biology, and there is actually an official reproducibility project in cancer biology. It's from the people from the Center of Open Science. What they do is they looked at some really cool papers which had a lot of impact in the field. They looked at some of those results, and they went to professional labs and said, hey, how about you reproduce figure 1b from this paper and figure 3c of the other paper? So they don't replicate complete papers, but they replicate key findings. And in particular, the people who replicated are completely independent of the people who initially reported the study. And then the idea was, you have a table and you just go through the top 10 papers in the field and you see how many of those were reproducible and how many are not. Simple story. Not everybody liked it. And you can easily see why the people who are in the list, who are under scrutiny, didn't like it. Because they were like, hey, I have the paper in the bag, right? Well, why are you guys now looking at this again, right? I mean, we did our job well. There's nothing wrong with us. I mean, if you don't get the right results, it's your problem, not mine. So there was lots of discussions about this. One of the most widely discussed reactions is from a researcher called Mina Bissell. Professor Bissell is at the, where is she? In Berkeley, one of these national labs in Berkeley. She's very famous for 3D cultures and very famous in the biomedical field. And she wrote a commentary in Nature about the risks of the replication drive. And she said the problem with things like this cancer biology reproducibility project are, if a researcher spends a long time in a highly specialized field and let me retry this. So if somebody from this project who is a replicator spends actually a short amount of time in a highly specialized field and then reports that what they found in the paper is irreproducible, this has an effect. So it can deter other scientists from pursuing this, jeopardize the original scientists' chances of getting funding, and potentially damage their reputation, which is correct. So if I don't want to have one of my papers in this list as being irreproducible, right? So what is her idea how we should do it better? She says what we should rather do, rather than this reproducibility project, we should just all come together and collaboratively solve reproducibility problems. which I think is exactly the right idea. We come to that in a second. But she got lots of flack for this. People like, you're an enemy of reproducibility, blah, 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 blah. And I think what came out is, why people reacted so strongly and why this was so widely discussed is, there is a notion of reproducibility being about the reproducibility police. So somebody comes and looks at you and says, I am the law, I reproduce your papers, or not. And it's a matter of life and death. Reproducibility is seen as a binary outcome, and either you make it or you don't. And if you don't make it, you're dead. You have like this black mark on your CV where one of your papers has failed. So we had Tim Errington, who is the lead of the cancer biology reproducibility project, as a guest in Cambridge a few years ago. And when he came, I pretty much used this to advertise his talk. I was like, come and see who are the good and the bad and the ugly in cancer biology, who is top and who is flop. And he saw these slides when he walked into the institute, and he was like, yeah, I see why you did this, but I wouldn't do it like this. And he gave this, in a way, very gentle presentation where he said, actually, it's not about top and flop. It's more about finding out what are the obstacles to reproducibility. If it's reproducible, everything's cool. If not, why did it fail? At what point did it fail? So in the beginning, he says, they were very strict. They were just doing binary, top or flop. And then they found out this was too strict because sometimes the only thing that went bad is that maybe the temperature at which they kept the cells was 2 degrees off. I don't know. And if you just changed this, you adapted it a little bit, everything worked fine again. But this would be counted as not being reproducible because it was not exactly following the recipe that was given in the original paper. So they then became much more interested in just seeing the changes. What did you have to change? How much did you have to change the recipe to actually still get the same result? And reporting all these obstacles rather than focusing on whether the originally reported thing directly, like when you crank the handle, gets you to the right result. So this is a very different view of how this reproducibility project should work. This is much more collaborative, just like what Mina Bissell said. This is helping each other, focusing on the obstacles, trying to see what the problems are in certain approaches and how we can overcome them. And if you want an example of how this works, so this is Mina Bissell again. She wrote a paper in Cell. She worked with Nelly Polyak, who's also very famous. Mina Bissell sits in Berkeley, Nelly Polyak sits in Harvard, and they both work on the biology of the breast tumors, the heterogeneity, different cell types in breast tissue. They are both world famous, they have superb labs, the best postdocs, the best students. They just faced this tiny problem that one of the labs could identify a certain subset of cells in the tissue which the other one couldn't. And they couldn't figure out what the problem was. So they had people flying back and forth, having a huge carbon footprint of just trying to find out what happened to those cells. It turned out, and that's the punchline of the paper, that one lab was shaking the cells and the other lab was stirring them. So shaken or stirred was the main difference. Everything else was the same. So it's a very minor thing, and I can't help but think that they learned a lot about their cells by just trying to follow up each other's work and seeing what the heck had happened to them and trying to understand what the real reason for irreproducibility was. So they were not shouting it, well, at least they didn't write it in a paper, but they were not shouting at each other saying, you are not reproducible, no, you are just too stupid to find them, no, no, no. They were just working together, and I think this is how it should be. And that brings me to an important thing, because I'm a computational scientist, so I think a lot about code and how to share my code and all how versioning, how the versions of different software, how to preserve that across time, how to use Docker and all kinds of computational things. But all the wet lab experiments have the same challenges. You have to describe what you do, whether you shake them or whether you stir them. And I don't think the same scrutiny, the wet lab notebooks are not under the same scrutiny as our code, and maybe they just should be. So the punchline is, embrace reproducibility. You will learn some new science by doing it, because you work with other people trying to understand what the obstacles are. So this brings me to reason number 6 of my 5 reasons to work reproducibly. The last part. Now imagine we do all of this. We all work reproducibly, we publish papers, we still care about the science, we work together on trying to find obstacles and trying to learn some new stuff. What do we actually do with, how do we update papers? if we find a minor mistake, what do we actually do about this? Now the problem is that scientific publications as we have them today are very static. They are written in stone or, well, I guess burnt clay. They are the final result. They are the static record of some scientific achievement, and they're very hard to change. And there is a mentality that you actually shouldn't look back. You have this paper, you just put it on your CV and you look forward, because the only thing that's really important is the next paper, not the last one. But sometimes you spot mistakes in stuff that you have published, and sometimes these are big and sometimes they are small. So in my own work I have several papers which have corrections. So one of them is, this is the paper which arguably got me tenure. It's my flagship paper, and there is this big red box when you open the paper, and it's like, this article has a correction. Like, I mean, if there's smoke, there must be fire, right? So something must be wrong with this paper. What is wrong with this paper? So you click there and you read it, and it says, the data are fine, the figures are fine, everything is fine. It's just that in the text, when we discuss one panel or we describe one panel of a figure, we say, this pattern is associated with worse prognosis, when we should have said, this pattern is associated with better prognosis. So there's one word is switched. It's a typo, it's a mistake. Everybody who looks at nobody can be mistaken when they look at the data or the plots, it just inverts the wrong word. It's a big red box. I mean, this is not a mistake, this is a little bug in the paper. Or here, just came out this year, we have this method paper, correction to this. What's this now? This paper has 5 figures, and in the first version they were in the order 1, 2, 3, 5, 4, and they should have been in the order 1, 2, 3, 4, 5. So this right figures just okay. And I think this, I have to say, this is a bit much. I mean, imagine how we deal with code. So because people think if there's smoke, there's fire. Think about how we deal with code. So places like Google, they will give you money if you find bugs, and they will not put a red button on the Google website, a red box saying, Google is flawed, a correction has been posted. I mean, they just fix the bug. And I think science should be the same way. If you find something as minor as one word which is mistaken, just fix the damn thing. Don't put red boxes everywhere. So, I mean, I think what science has to embrace is that everybody makes mistakes. And if they are major, okay, like the people with the fish, right? If you can't find your data anymore and your laptops and all of this is big, there must be expression of concerns, there must be correction and retractions. I think we sorted out how to do this. But what if you have really minor mistakes? So one of my friends in Cambridge has the problem that one of his students stupidly went back to 2 papers they had published a few years ago and looked at the code again. Young and naive student, why did he do that? So he found that very early on they had used the wrong scaling factor for their data. Nothing really was changed in the end results because the scaling affected all the data and they only looked at relative frequencies, but for some of these genes something changed. So they went to the journal and, well, they first talked to their collaborators, biologists said, did anything change? They said, yeah, actually pretty much every number in the paper changed. But he was like, but did the biology change? No, biology is the same. It's just instead of 300 differentially expressed genes, we now have 325 differentially expressed genes. I mean, who cares? So collaborator said, well, that's minor, it's just sweep it under the rug, right? But this didn't sit too well with my friends, so they actually went to the journal editor and they explained the problem. And journal editor thought, oops, we're in this scenario here. Every number in your paper has changed. Oh my God. They were like, no, but the story is still exactly the same. It's just really I mean, nothing has changed except that everything has changed a bit. So they had a really long discussion, half a year of pain, and the student didn't feel really well. I mean, he shouldn't really have checked, honestly. So I think what's needed is there need to be some new ways of how to dealing with, like, treating publications being less static, having an idea of maintaining a publication rather than just keeping it there in cold storage. And the idea must be something like this. You have an initial study which leads to a publication. People are really nice. They have open data and open code, so people, including themselves, can reproduce the study. And now there must be some smooth and transparent ways of updating a publication. And you can think of, I don't think this is really implemented in any journal I know of, but what it would entail maybe is a new type of submission. So you go to the journal webpage, it asks you what do you want to submit, an opinion piece or a research article. You say, no, I want to submit a correction. And it just says, you explain what happened. You said, well, this one scaling factor was off, and now we have 325 genes and not 300 genes like we reported previously. And then there needs to be some editorial process. The editor needs to look at this, see how big the change really is. Maybe, if it's bigger, potentially re-review this particular part. Why not? Journals know how to send things out to reviewers. And then there is a decision, and you just have a new version of the paper. You have a bug-fixed version of the paper. And some journals already have publication histories for, I took this, I think, from eLife. It tells you when the paper was received, when it was accepted, it tells you when the version of record was published. So the only thing I'm asking is, now please also keep track of minor amendments. And just have, just like if you go to archive or bioarchive, you just always get to the newest version of a paper. Let's do the same for real publications. I think 1000 Research does it, eLife is talking about it, I think PLOS CompBio also keeps track of things. So some journals already do, but it is not an established way of how people think about their papers and how we act. And that brings me to reason 7 of my 5 reasons. So except for avoiding disaster, finding it easy to write papers, easy to talk to reviewers, the continuity of your work and your reputation, and the fact that you learn some new science by working with your colleagues, you should also do reproducibility to keep your papers bug-free, because papers with less bugs are better papers. So, simple question. When should you, all of you here, worry about reproducibility? Well, before you start the project. Because there are skills involved, skills you need to learn. There's things like Git and R Markdown, and there's an investment in time, and you have some technical skills you have to learn. Do this early on. While you do the analysis, because if you forget about it, you're in the same situation like my postdoc who forgot how to report the parameters for his network, and then we didn't publish the paper. When you write the paper, because you want the newest numbers and the most accurate numbers in the paper, when you co-author a paper, because I would not trust anybody, and you don't want to be in the situation of the co-authors of the Duke guy. After you've published a paper, because if there is a bug, you actually want to find it and update the paper. And when you review a paper, so check for whether the code is available. If not, ask for it. They will give it to you. You're very powerful as a reviewer. You just turn into ugly reviewer free if they don't collaborate with you. So in short, when should you worry about reproducibility? Always. Now, who should worry about reproducibility? Well, there's the people on the ground and the people with the vision. The people on the ground, they need to learn the tools and apply them in daily work. So they need to worry, all the students, the trainees, the postdocs, research staff, all of them. The PIs in a way play a different role. I mean, we have all stopped doing real work a long time ago, and now we just survive on talking about other people's work. But the role of the PI is important because in your own lab, you're the boss. So you have to kind of set an agenda, reproducibility agenda, and build up an environment in which reproducibility is being valued. And you have to create a culture of reproducibility in your own lab, in which all of these people can thrive and actually they feel rewarded for using all these tools. And then, given what I told you, your science, your career will be even better. That's it. Thank you very much.
So I would now like to introduce, thank you so much, Florian. I would like to introduce Rob Hoft, who is the program manager and the guru for research data at the Dutch Tech Center for Life Sciences, who will be chairing the Q&A session and also giving the closing remarks. So, welcome.
So we'll have to throw the microphone. So, any questions for Florian? There must be lots of questions. No? Well, then I have the privilege of asking you the first question. So, my big hobby is helping researchers with data management planning.
Yeah.
And do you have experience with data management planning, and how do you connect it with this?
What is data management planning? Is this little paragraph I write in my grants where I say where I store? Yeah. I think that's the experience I have with that. Sounds very tedious. So what do you tell researchers?
Okay, so now I take the floor and tell people my story. So I think I'll have the chance of giving a little bit of that in the closing remarks. But
Yeah, I mean, I don't have anything really smart to say there. I mean, except that, I mean, given the way we work, I can point to a lot of a track record about how we share data and the repositories we use, how we share our code, and I have lots of evidence that people reuse our code, look at our data. Apparently all the grant funding agencies are happy with the way I write my data management plan, so that's the only... I never got feedback on it, so nobody ever looked at it and said this is good or bad, they just gave me money. So.
Very, very good. Okay, other questions?
Alice Dunning from the library. I'd like to go back to that example you said of the scientists from Berkeley and the scientists from Harvard who'd the shaken not stirred, right?
Yeah.
They did an awful lot of work to discover something that is really, really minor.
Yeah.
Right? Was that really worth it? And is it really worth all scientists who are working together spending that much time to explore the shaken?
No, the reason was minor, but the effect it had was major. So there was one lab who just couldn't identify these cells. So the debate was, what are all the different cell types in this tissue? And one lab said it's A, B, C, and this one type, and the other one said we only find A, B, C, we don't know what you're even talking about. And it's right, in the end, the reason why they couldn't see it was minor, but the scientific insight was the difference was big.
But is that always likely to happen, right? Because it could be the reason why two experiments aren't reproducible is a very minor contextual issue that isn't about science. You know, there's more vibrations in one lab than another. Another lab has a different lighting. And do you really want to go all that time to discover that there's...
I think people do that. I mean, they're very frustrated by exactly these things. I think this is science for most people. There are lots of stories. I know this in Cambridge from people moving from one building to the next, and their mouse models are no longer working. I mean, if nobody knows why. They just ignore this because it's too hard a problem. Curing cancer first and then figuring out why in one room it works and the next room it doesn't. But I think they would learn a lot if they had figured this out. I mean, some of my colleagues, we work with estrogen receptor positive cells, so they respond to some hormone, and sometimes the experiments no longer work because this particular cell line just doesn't do its job. Nobody knows why. And it might be something really minor, but I think the scientific insight and understanding what actually happened would be massive. And people are just ignoring this right now. Because it's something, it sounds very technical, it might be a minor issue like shaken or stirred, and you better ignore this. Better move on.
Thanks, Alistair. Other questions? Okay.
First of all, can you hear me? Yeah.
Yes.
I really recognize what you're saying because that was exactly what science was for me as a wet lab biologist. Everything can matter, and then you can spend years even to try to get a procedure work. And my question to you is where to start? Let's say you now today you've joined to this talk and you want to start somewhere as a PhD postdoc or as a PI. What would be your recommendation?
I think the first thing, okay, I took this out because the talk was too long already. I have a section on baby steps. The first thing we teach people, I try to teach people, is what a concept called tidy data. And tidy data is, so who has a PhD here? So how did your PhD thesis, can you remember how this was called? For most people it's called my thesis version 2, my thesis version 2 final, my thesis version 2 final, really final. This is not tidy. Right? This is just really everything meshed together. So it's more about how to represent data in a way it's easily read in by a computer, about naming standards. These are really simple things, but they improve the way people can understand your data and your code massively. And I think that's the first thing we teach them. And the medical school in Cambridge is now teaching basic data handling skills on a computer. Like most of this is like R and just look at file names and not having blanks in file names and all kinds of stuff. It's really basic, but it has a massive impact, I think.
Thank you very much. I'm Han Heijmans from the TU Delft Library. I always hate it when I completely agree with the person ahead of me, because then I don't have any serious questions to pose. So that leads me to taking the opposite position. So you very rightly talk about a very competitive field science is. So many scientists have an advantage because they have special methods, special instruments, and they actually want to keep that secret. So how do you react to those scientists?
Yeah. As I told you in the beginning, I'm not a preacher. I mean, live and let live. You do your science the way I do. My attack point is I point out how much better they could do this job and how much more success they would have if they actually work reproducibly. The way to get onto, I think it's, I would use the ability I have as a reviewer of papers to just request data and methods and all of this out in the open. And I flag this up when I read papers. I flag this up when in talks when papers are published and the data or some key component is just not well explained. This can be flagged up to the authors, to the editors. I'm just very annoying when it comes to this.
Follow-up question.
I think if you look into history, then you see that, for instance, Dewar and Kamerlingh-Onnes in Leiden, they were very competitive of getting the coldest spot on Earth, and it was all technology driven. So they invested lots of money in building equipment, in building up expertise from their crew, etc., because the science was so competitive.
Yeah, yeah.
So I hope you agree with me that science should be less competitive, that would help.
Yeah. There's also a special situation there, I guess. I mean, I know that my science is less competitive than that. I mean, I've never been scooped in my life. I mean, people talk about being afraid of being scooped and all of this. I'm like, they're just too much boxed into one particular thing. If you take a step back, you realize, well, there's other papers in the same area. It's not really. I mean, there's always space for another paper. And there's just people are very boxed in, obsessed with some things. I think actually for me, it's all about the niche, and I have my little niche, and it's not, I've never been in a situation where I was head to head with one other lab and was like, who is faster, and then you think about how to be unfair and hide and stuff. It's never been like that for me, and I would hope that most science actually is not. It's just these stories are so exciting when people really fight, right? Yeah.
So, thank you, Florian. Thank you for the questions. Yeah, I'm also very much agreeing with most of, almost everything that you said. So, of course, I have to...
So it's a very boring talk, that's what you said.
This,
I mean, preaching to the choir, everybody already convinced.
No,
I mean,
no,
good,
no.
yeah.
So, I can tell you why I was triggered by the title of your talk. So, my interest is really in data management planning. I found this a fascinating story on how to get to good papers and reproducible papers. For me, the data is the key. I find computer readable very, very important next to the human readable papers. And I was very happy in the end when you said you need to always be reproducible. Because I think that reproducibility doesn't even start early in the project, but as I said, just said, the data management planning is actually where you plan things like what you're talking about in the naming system of files. You plan how you are going to make sure that you will be reproducible in the end. And this is something where a data management plan request from a funder actually helps to trigger this in a lot of researchers. The problem with that is then that the researchers actually see this as a burden. And this is exactly what you're telling me again for the reproducibility. The researcher sees making a data management plan as a burden, but actually it's an investment. You show excellent statements on how this actually helps. And these reasons are going to help me really as well to convince researchers that it isn't a burden, it's actually a benefit. It's an investment that you're making your own research. So, reproducibility for me is the first step towards reusability, and I am one of the 54 authors on the paper on fair data, and so author number 27, right in the middle. Reusability is the fourth letter, and I think reusability requires reproducibility and a little bit more. We're actually soon organizing as DTLA a meeting on notebook standards and how to actually collect enough data, metadata, to make reusability possible. I've talked to people who were adamant about not having any problem opening their data because nobody would ever be able to use it anyway. So that's of course the one thing that we're trying to avoid with fair data. Right. On the other hand, I've also met people that said, I don't want my data to be out there because if my data is out there, other people will start using it and they will come to me and annoy me with their questions. Well, same thing. If you're reproducible, if you document your data well, I think you can avoid those questions and still help science forward by allowing people to build on your results. So I am very happy you built a nice castle that I will build on top of for convincing people to make fair data. Thank you.
Thank you.
Thank you so much. And just before you disappear, I would like to have a couple of thank you words. So first of all, thank you for our today's speakers, Florian. So please join me in applause again. And I would like to thank Rob for chairing the Q&A and also for the wonderful and inspiring closing remarks about the importance of good data management and making sure you keep your notes of all your experiments and make them reproducible. So thank you for that. And I would like to thank everybody from the library who made this event happen. So not only sponsoring today's event, but also helping to organize it. So thanks everyone. And to all of you for coming, of course, and participating in these discussions and joining us today. So thank you very much for coming today. Okay, and finally, I also wanted to let you know that there will be another Future Forward seminar series, and this time we'll have an internal speaker. So there will be Professor Bernd Rieger from TU Delft Faculty of Applied Sciences, and he'll be talking about why he thought it was important to share imaging data. So all the microscopic images he's taking, how does he do that, what are his motivations, and what are the challenges. So hopefully you'll be able to join us. It will be on the 10th of December, so please book the dates and we'll send reminders afterwards about that event. And just a reminder, if you haven't yet filled in the feedback forms, just make sure that you indicate your choices to let us know what did you think about today's event and return them to that basket down there before you leave. And before you leave, very importantly, to facilitate discussions, conversations, Florian will be able to participate in the drinks together. So please join us for drinks. Drinks are hidden, but they are there. So we have some wine in there and there is a fridge. So actually we made sure that your drinks are really nicely chilled so you can have a cold beer from the fridge. So please join for drinks and thanks again for participating in today's event.